whiteboards out and draw it together. So let's get started, and we're going to talk about fungi. So it's our first fungi day, and do we need the keys? Is that door locked, you know?
Yeah.
Somebody should lock it. Okay.
Okay, I just need to take a look at it.
Thank you. Okay, so we're going to talk about fungi today. So everything gets a little bit strange and gets a little blurry between the different organisms. But the cool thing about fungi is they're nothing like bacteria and they're nothing like viruses. So we've moved on to eukaryotes. We're talking about eukaryotic organisms. And I think it's important to talk about fungi, especially because it's been in the news so much. And why do you guys think it's been in the news so much in the last 5 or so years? Totally wild, crazy question.
Because The Last of Us.
I'm totally gonna give you a candy for that one. That is true. Yes. My husband always talks about that. I forgot about that. Yes. That is totally wise. So in The Last of Us, what happens? They get like a brain?
Yeah, there's like a fungus that's able to evolve to withstand like the heat that the human body usually uses to kill it off.
Uh-huh.
And it kind of, like fungus that goes for insects and kind of makes a little zombie insect.
Yes, I have videos about that
Scale
too.
of humans.
Oh yes.
Creates little fungi zombies.
Yeah. Oh, awesome. Yes. So I do have a video about that today, but I did forget about that show. My husband talks about that a lot. Awesome. If you think about fungal organisms, you think like you're walking through the forest and you see mushrooms and you see fungi growing on stuff. Like, why do we start worrying about it more and more? It's a totally random question, so if you have no idea what I'm talking about, I don't believe it. But more like the contemporary worries around fungi. About some things. I don't know, I'm asking it to you in a weird way. But what we're worrying about right now about fungal infections is because of climate change. So the world is shifting temperatures between different zones that are not used to being so hot or not used to being so cold. So we are really worried about fungal infections, and fungal infections especially for plants. So our entire crops all around the world that's our life source. And if people eat meat, it's the life source for the animals. And if it's our entire population is vulnerable to how well our crops do. And just like the Irish potato famine when was that? Does anybody know offhand? It was in the 18 something. I can't remember. Yeah, Google it for me.
I feel like it was the late 1790s, because that was in a show I used to watch called Outlander, and they talked about it.
I love Outlander. Let's talk about shows.
1845.
When was it?
1845.
1845.
Oh, I lied. Sorry.
Oh yeah. So 1845. So the Irish potato famine. So it's a significant portion of the United States population has some sort of roots in their Irish ancestry. And that is because of the Irish potato famine. So thousands and thousands and thousands of people left England and Ireland and all of Great Britain, left Europe and came to America through Ellis Island and to America because they were starving to death, because of the potatoes had a fungal infection, and because of that fungal infection, the entire crop of all of Europe basically was rotten. And so people were literally starving to death. And I know, don't like everything I talk about, I scare the crap out of you, but it's really not that far off, with our population, the climate change and the population growing and the situation with our climate we've gotten to a place where it's unpredictable, where we're not 100% sure how our crops are gonna do. And if there's a fungal outbreak, then what are we gonna do about it? And how do we feed this giant population that we have around the world? So yeah, it's a huge area of research right now. And there's another reason I'll scare you, but I'll wait till later. Yeah, go ahead.
From what I understand, part of the issue is just that the crops are too uniform. Like,
That is
for
a
example,
huge
the
problem.
bananas. Like, what was it, 20 years ago or 50 years ago or whatever it was, there was an illness, I don't remember what caused it, but an illness for the bananas, and the type of banana we used to eat no longer exists because it got completely wiped out.
Yeah.
And so now we're eating a different kind, but the kind we're eating now, they're always the same. So that's the problem. If we would just farm more variety, then if an illness came in, like if we had lots of different kinds of potatoes and lots of different kinds of bananas and like all the stuff we were growing had more genetic variety, then it wouldn't be so vulnerable.
And I 100% love that discussion so much, because we're gonna exactly address that same problem when we talk about like antifungal resistance and sexual reproduction between fungal organisms and why we worry about that. But that is a great point that Dr. Meyer said, is that we are genetically modifying, and even it's not really considered genetic modification if they're just naturally breeding crops to where they're homogenized so much where there's not enough mutation going through the population. So that is actually, I think I was 14 or something when I first learned about the cheetahs, and the cheetah population in the world, they're basically all inbred, where so much so that you can give any, do like organ transplant or skin transplant or anything between cheetahs of any population anywhere around the world, because they're so genetically similar. And so if there's a virus that runs through or some sort of genetic breakdown within that population, then cheetahs are gone and there's nothing we can do about it because they're so interbred. So yeah, I think that's a fantastic point. And yeah, so we do have lots of problems. Fungal biologists are becoming more and more vital around the world, and they already are absolutely with crops and plants, because our crops and plants can't survive without fungal organisms, but also with medical mycology, it's called, with medical mycology, because the human population is extremely vulnerable to fungal infections, but we'll kind of go into that and what's going on there. So the last story I'm gonna break out is that, personal story, because this always reminds me when I first started talking about it, is this is what I was learning about in undergrad, and he was my boyfriend at that time, we're married now, but we were sitting on a park bench and I was telling him all about mycorrhiza, and I was like, mycorrhiza are so cool and this is why, and it's like this, there's now a lot of research about it actually, about how trees and plants and the entire population of organic material is connected underneath the earth by fungi. And so I was telling him all about this and he wasn't listening, and I was like, listen when I talk to you, and then he proposed to me right there, and I was all, oh, that's why you weren't listening. So now we always have this like mycorrhiza joke. So every time now I'm like, listen to me when I talk to you about mycorrhiza. So yeah, I think I was like fascinated by this. I think it's a really neat topic, and yeah, so we'll learn a lot about fungi. So people say fungi, people say fungi, they're all correct. It depends what country you live in. You decide how you want to say it, it's totally up to you. Okay, so I want to save talking about mitosis and meiosis till the end, actually. I was going to go through it at the beginning, but I want to talk about it at the end, and the reason is because I want you guys to draw it out yourselves. I'll get the boards out and you guys can draw it, so then you're actually practically doing some hands-on stuff. And then I have a coloring page for us on Thursday too, which I need to remind myself. So the coloring page that I have for us helps us to understand fungal reproduction, because fungal reproduction is crazy. And so it'll help you kind of organize yourself of how they reproduce. Coloring page. Okay. So here we are talking about fungi. We're in our special phylogenetic tree. Now we know what our eukaryotic organisms are. They're over there. They have organelles. They have all the organelles. See if you can name the organelles and the functions of the organelles. We start with more complexity in our division of labor, but don't forget, this can be one single-celled organism. That can be one organism that goes about living its life, or it can be part of a greater system that's a multicellular organism that live together, that have to function together. Yeah?
Does the tree say that we're more closely related to fungi than plants?
You know, I would not be surprised, but it does, well, it says here this breakoff is the same
At the same
breakoff.
time?
Yeah,
Okay.
if we would be further up here, we would be more connected, but here is the similar breakoff.
Okay.
Yeah, which, you know, is a little bit surprising to me that we're not more connected with amoebas, but that we are more associated with plants, but yeah, I don't know, I love talking about evolutionary biology too, but I don't know very much about it. Okay, okay, so, oh, this is the movie I was gonna show you, and this is what Dr. Buglino was talking about as well.
In jungles, few are more successful than the ants. There can be 8 million individuals in a single hectare. But jungle ants don't have it all their own way. These bullet ants are showing some worrying symptoms. Spores from a parasitic fungus called cordyceps have infiltrated their bodies and their minds. Its infected brain directs this ant upwards. Then, utterly disorientated, it grips a stem with its mandibles. Those afflicted that are discovered by the workers are quickly taken away and dumped far away from the colony. It seems extreme, but this is the reason why. Like something out of science fiction, the fruiting body of the cordyceps erupts from the ant's head. It can take 3 weeks to grow, and when finished, the deadly spores will burst from its tip. Then, any ant in the vicinity will be in serious risk of death. The fungus is so virulent, it can wipe out whole colonies of ants. And it's not just ants that fall victim to this killer. There are literally thousands of different types of cordyceps fungi, and remarkably, each specializes on just one species. But these attacks do have a positive effect on the jungle's diversity, since parasites like these stop any one group of animal getting the upper hand. The more numerous a species becomes, the more likely it will be attacked by its nemesis, a cordyceps fungus.
Awesome. Okay. So very cool, right? I don't know if you've
Incredible.
ever seen this show, but it's I think it's so crazy. Okay, so yeah, fungi are super cool. That it's maybe not that far off that humans can get an infection, a fungal infection in their brain, which actually people can get a fungal infection, get meningitis or actually sepsis from fungal infection. So we'll talk more about that.
Why do people have that?
You know, it's whatever part of the brain that the fungus is infecting makes them want to climb up, which is so genius of the fungi that, because then the spores will rain down on the rest of the, isn't that crazy? So crazy, total trip. Okay, so there are 5 phyla within the kingdom of fungi. We're not gonna remember those. We're not gonna memorize any of these because a lot of them are non-pathogenic. So we're just going to focus on the specific genus and species that are pathogenic to humans. So we don't have to memorize any of this. And we're just going to go through the individual fungi that cause infection. So it's a good review. Kings play chess on fine grained sand. We didn't talk about that yet, did we? Oh no, we did. We did at the beginning. Yeah, we talked about genus and species and kingdoms and domains and phylum and all that kind of stuff. So it's a good time to review if you need to go back over that. Okay, so this is just an overview, so you guys can read through that on your own. I sometimes like to do a little synopsis. But let's go into fungal morphology. So this is, remember, morphology is like the representation, like a kind of a visual representation of what that organism is. So if you talked about colony morphology in the lab, it's what does that colony look like, is a fancy way to say it. So we have 2 different types of fungi that we talk about, yeasts and molds. And even though they both belong in the fungal kingdom, they're both very, very different. But they look really different, but actually some of the characteristics about them are the same that we'll go through, so you'll be able to see why they put these 2 seemingly non-related groups into one group called fungi. So yeast, these are yeast, what they look like under the microscope. These are molds, what molds look like under the microscope. These are both computer kind of animated, but so yeast, such as Saccharomyces cerevisiae. So Saccharomyces cerevisiae is the yeast that you pour out of the packet and put into bread. It's also, they make beer, some types of wines are made from that. And so maybe that gives you a little inkling about what we're going to talk about when we talk about metabolism. But some species of fungi live in this life and they always look like this. Some look like this. And then we have another type that are called dimorphic fungi that have 2 different morphs, 2 different shapes, and they can be either yeast or mold, traditionally depending on temperature. So I'll go through that. Okay, so here are molds. This is what molds look like. So this is traditionally what you would think is like the stuff that grows on strawberries or whatever. This is fungi, a fungal infection that grows in mold form. And you might not always know automatically without using a microscope if it is a mold. Sometimes you have to have a microscope because, like, for example, athlete's foot. Somebody that has athlete's foot, you don't see, it doesn't look like a mold, but if you looked at it under a microscope, it does look like this under a microscope. So you'd have to inspect it, do some microscopy work to be able to diagnose your patient correctly. So here are molds. This is what are called hyphae, and hyphae or hypha, hyphae is fine. So hyphae are individual branches. Mycelium is what are a bunch of branches together. So we have 2 new terms, well, kind of 3 new terms, 4 or 5 new terms. Molds look like this, yeasts look like this. Dimorphic fungi can do either or, depending on temperature. We'll talk more about that. Hyphae are branches. Mycelium are a bunch of branches all together. And then when you talk about yeast live individually like this, but sometimes yeast trick us into thinking they have hyphae, or they might trick us into thinking they're a mold because they have this weird thing that they can do called pseudo hyphae. So pseudo is like false. They have like false hyphae where they look like they're in branches, but they're not. So you'd be able to tell if you zoomed in on the microscope if it's pseudo hyphae or traditional hyphae of a mold, but that's sometimes what yeast do. And then dimorphic fungi, you might look at the microscope and it looks like that. You might look at the microscope and it looks like that. So it depends on the temperature if it's a dimorphic fungi. So let's talk more about yeasts. So this is what makes yeast unique. So what makes bacteria unique? Everybody really quick, 100% attention. What makes yeast unique? I'm sorry, no, not yeast. What makes bacteria unique? Peptidoglycan. So there were a handful of people that got that wrong on the midterm, no big deal at all. Make sure you 100% know that. Peptidoglycan is unique to bacteria. Bacteria are the only ones that have them. And so now we're gonna talk about what make fungi unique. So yeasts and molds both have these unique structures, and they're called manoproteins, glucans, and chitin. But they're not specifically unique to fungi, but let me tell you about what I mean. So manoproteins, it's a mano-sugar with proteins together. So a glycoprotein, glyco means sugar, protein means protein, so it means sugar proteins. So don't get tripped up on the chemical structure. It's not that important. Just know that they have manoproteins. Those manoproteins are part of the cell wall. So do humans have a cell wall? No, humans do not have a cell wall. Bacteria do. Bacteria cell walls are made of peptidoglycan. Fungal cell walls are made up of these three things. So they're made up of manoproteins, which are unique to fungi. Yeah.
Is chitin the same stuff that makes the exoskeleton of bugs?
Yes, yeah. So yeah, let's get to that. Yes, good question. So glucans, what glucans are glucose. It's a polysaccharide of glucose molecules. So they are unique to fungi, but they're also unique to other plants. So if we're staining an infection and we're trying to differentiate between a yeast infection and a bacterial infection, bacteria have peptidoglycan, yeasts and molds have glucans, but also just know from the top of your head that glucans are not just unique to fungi, plants have that too. But would your patient have a plant infection? No. So we can just say they're unique to fungi as a pathogen. And then chitin, the same thing. So chitin is what also insects have chitin in their exoskeleton. And what chitins are remember we talked about nags and nams make up peptidoglycan. It's acetylglycans and I can't remember. Whatever. So nags and nams make up peptidoglycan, but these are nags individually. So it's like glucosaminoglycans, something like that. I can't remember 100% what they're made out of, but all we need to know is chitin. Chitin is unique to fungi compared to bacteria or other pathogenic organisms, but some plants have these and insects have chitin as well. So these are what make pathogenic organisms unique that are in the fungal domain. Yeah, are you guys with me? So, I'm sorry, go ahead.
Did you say that not all fungi have chitin?
No, they all do.
They don't do.
They all do, yeah. So bacteria, fungi. Just keep a running list. Peptidoglycan, manoproteins, glucans, and chitin. And then we're gonna do all of our organisms that way. Is there something that comes to mind? I'm gonna ask you another. I hate when professors ask random questions that nobody can answer because they don't understand what they're asking, but I'm gonna try it and see if I can get you to understand it without giving you the answer. So what sticks out to you when you talk about the uniqueness of fungi? Does anything kind of trigger your thoughts of like, okay, my patient has a fungal infection. What is triggered in my mind when I think about the uniqueness? Yeah, sorry, go ahead.
How do you treat it?
Yes, how you treat it with what? Yes, with antifungals. Awesome. So we're looking for drugs, for pharmaceuticals, and when you think about those targets, that's really important, is to think about the targets that make that organism unique so that we can think about, okay, if we're gonna target a fungal infection, how are our pharmaceuticals gonna work against that infection? Yes, awesome, great. Go ahead. Okay, so here's our cell wall structure of fungi. We have a couple other unique things. Oh, the last one I wanted to talk about was ergosterol, but we'll talk about that later on too. Let's add that to our list because ergosterol is specific to fungi. I should add that to my slides. Ergosterol. I will talk about it, but ergosterol is a type of cholesterol. So animals have cholesterol in their phospholipid bilayers. So it's a type of lipid, and there are lipids in our phospholipid bilayers that are called cholesterols. So we have cholesterols in every single one of our cell phospholipid bilayers. Animals do, and humans. So that's why people who eat a lot of animals get high cholesterol, is because they're eating it. So same with fungi, they have something that's called ergosterol that is similar to a human cholesterol, but it's unique to fungi. Okay, let's move on. So the internal structure, same. They're a single-celled organism. Yeasts are single-celled. Yeah, go ahead.
Do they have a glycocalyx?
Do they have a glycocalyx? They do.
Oh, the meaning of it.
They do, but let me look it up because I'm not 100% certain how that varies between yeast and molds. Yeah, let me look that up because that is interesting. Good point. Because a lot of eukaryotic cells do, but I'm not 100% sure how that works in yeast versus molds. Okay, so here we are with a yeast. This is what it looks like when you pour out that packet and you are making bread. This is as one of those single cells. So one of those single cells is an individual yeast, and they have the exact same organelles that we do. So their cells have Golgi, they have rough and smooth endoplasmic reticulum, vacuoles, mitochondria, ribosomes, vesicles, all the same stuff that we do. This little thing here is called a bud, and we'll talk more about that later. So basically what it's doing is asexually reproducing, but they asexually reproduce slowly, and that little bud grows and grows and grows, and then will split off. So we'll be able to see that in the microscope. So if you compare the internal structure of bacteria, yeast, and an animal cell, I just liked to look at this. I thought this was kind of a cool visualization from, I got it off Google. So then you can compare between a prokaryotic cell and two eukaryotic cells. So colony morphology, when you look at yeast on an agar plate, they look different than bacteria. They look, I mean, kind of similar, but they're bigger, they're creamier, they're really shiny, and they grow like crazy, but they also require a lot of sugar. They require a lot of agar, so they will work through that, whatever nutrients are left on that agar plate pretty rapidly. So that's what they look like on an agar plate. We'll grow them. You'll be able to see them on your own later on this semester. So microscopy. So this is, my sister actually taught me this. I talked to her a lot about the clinical side of microbiology because she's a physician. And so I know the research side and she knows the clinical side, which I think, you know, the clinical side is more pertinent to you guys. So I try to ask her as many questions as I can. And so when your patient comes in and you think they might have a fungal infection, physicians call it, or any kind of healthcare practitioner calls it spaghetti and meatballs. And I know it sounds gross, but that's what they call it. If it's spaghetti and meatballs, it's a fungal infection. That's what they say. So but don't forget that these here, to me, these look like pseudo-hyphae. These don't look like real hyphae because they're not, there's no mycelium, they're not really branched. And if you zoomed in a little bit more, you'd be able to see individual cells and them kind of coming together. But just don't forget that some are only the meatballs, some will only be the yeast. But many of them are either dimorphic, or are molds, or have some sort of pseudohyphae. So you'll see this a lot. You'll get what I'm saying as we move along in the fungal section. Don't stress about it too much right now.
So,
yeah.
The spaghetti and meatballs, is that dimorphic then? If you've got both in the same, are they just two different yeasts and molds together?
That one would look to me like it's just a yeast. It's not a dimorphic. So just yeast are able to also have
So that's the pseudohyphae.
pseudohyphae.
Okay.
Yeah. So yeast can have pseudohyphae as well. But you won't have to identify it in a microscope for now. Later on in the semester you will, after we do it in the lab, and you'll be able to see them in the lab. So one thing that we will ask you about in the lab later on when we get to that section is when you're looking at it under the microscope, here's a microscope at 400 times. Here's a microscope at 1,000 times. And when we look at Staphylococcus aureus, for example, they look like about that size. Where if you look at yeast, they're like this size at 400 times. So, you know, have you ever looked in a microscope and ever seen anything that big in this class? Like, we don't see anything that large. And if you notice it's a big, large thing, it's gotta be something that's not bacteria. Remember bacteria and blood we look at under 1,000, under the oil microscopy. Everything else, we don't need it. Okay, so we'll talk about that more later when we get to the lab portion. But just so you know, spaghetti and meatballs, that's what physicians and healthcare practitioners say when they're looking at the microscope. They're like, yes, it's a yeast infection or a mold of some sort of fungal infection. And we'll talk about how to identify them also in diagnostics. Okay, so let's talk about molds. So molds are also called filamentous fungi. So don't forget that because you'll see that a lot of times in literature or in a clinical setting. You'll see filamentous fungi, the molds described as that. So here's mycelium, here's a hyphae, here's the manoproteins, here are the glucans, this is just a cartoon. Here is chitin, here's ergosterol, that's little like cholesterol that's within the phospholipid it's not actually within the phospholipid bilayer, it's within the chitin layer, but sometimes it can be within the phospholipid bilayer too. Okay, so let's look at cell wall structure of a mold. So they have chitin, manoproteins, glucans, same as yeast. So they can form these septum. So these septum are whether they can come all the way together or they can stay apart. So it's almost like how we talked about Staphylococcus aureus or Streptococcus pyogenes. When it's Streptococcus, it's like this, right? And those are all individual organisms. But here were the septum that they don't actually physically separate. So The same is true for a mold, but the mold itself, even though if they have full septate hyphae, this is all one organism, this is 4 organisms. So the septum, they might not separate all the way, the septate, or they can separate all the way, but it's still one organism. Does that kind of make sense?
Why do they consider it one organism?
You know, that is a good question, because this one piece, which we'll talk about when we get to repro, this one piece can break off and create its whole own organism, and can branch off and create its own organism. But, I mean, maybe because they share nutrients and they move together, reproduce together,
But
I'm
don't
not
the
sure why.
bacteria do that too?
Not, well, but this one,
Sorry.
no, that's a good question. I would guess it's, yeah, I don't know. Good question, yeah, all good thoughts. Love it. Okay, so the cytoplasmic membrane that has ergosterol in it, it's a type of cholesterol. And then here's one term that we haven't talked about yet that's called a Spitzenkorper. Spitzenkorper. So a Spitzenkorper is this little end piece right here that has these digestive enzymes. And these toxic enzymes, what they're gonna do is they spit out these toxic enzymes in the end to degrade the material around it so that it can keep moving and branching out and branching out. So that's what a Spitzenkorper is. And if you can imagine, when you look at a fungal growth like in a tree, like it can grow through wood. They can grow in all kinds of different rocks and crazy, really strong structures because of those enzymes that break out at the end. But what does that make us think about when we talk about human health? That's terrifying, right? Like, so a fungal infection, if it can break through a rock, and then it can also break through your tissue and your lungs and things, or your patients, I don't wanna say you. So Spitzenkorper, they have these toxic enzymes that we'll talk more about later on, but that's what it's called, is a Spitzenkorper that's at the end. Okay, so more on their internal structure, they have mitochondria, vacuoles, all blah blah, all the same things. They have these little ergosterol crystals within their, this is number 5, they're just these generic little cartoon, but these little crystals that give their hyphae strength and rigidity. But other than that, they're all the same. So hyphae, there's 2 types of hyphae that we call it in clinical medicine as well as in nature, are vegetative hyphae and aerial hyphae. So vegetative hyphae are hyphae that dig down and into the substructure, into the substrate, and then aerial hyphae reach up into the ether, and so those can reach up into the lungs, or they can block tissues and things like that. But those aerial hyphae, the point of the aerial hyphae is reproduction, these reproductive structures at the end that we'll talk more about. Okay, so I'm just gonna play this one for you, but this one that doesn't have any sound, I just want you to get a sense when we're talking about fungal spores. Just get a sense for this video really quick. So this is like, it's not under a microscope. This is just lighting, how it works with the camera. It's just zoomed in. But these are fungal spores. And so like mushrooms, for example, or different types of hyphae, different types of fungi can produce millions, billions, trillions of spores. And every single one of those spores has its entire genome in it. So the DNA that's inside of one of those spores is enough to go and plant and make its own fungal organism. So when you're imagining treating your patient with this type of infection, it's a little little terrifying, right? Like, how do you prevent the spread of a fungal infection if your patient has a mold, has some sort of filamentous fungi infection in their skin or in their lungs, and how do you prevent it from spreading? How do you treat it without harming your patient? So that's just, mind-blowing to me. I thought that was kind of cool to bring up. Okay, so we're gonna talk more about spores and how they reproduce on Thursday, but we're just gonna introduce it a little bit today. So, let me see how. Okay, so there's two types of spores. One are called conidiospores. Wait, I think I passed up the first one. Oh yeah, here's sporangiospores. So we start talking, our first type that we talked about are sporangiospores. And how I remember this term is because it has the word spore in it twice. So a sporangiospore, it kind of sounds like spore. Sporangiospore has spores within the spore. So that's how I remember what it looks like. An example of those are called rhizopus, and we'll look at those in the microscope later on. But it's a spore within a spore. And then we have conidiospores that can look like paintbrushes, talking about Outlander. Do you remember that part? And then, or they can be, the conidiospores can not have a vesicle or have a vesicle. Let me show you what I mean in a second. So conidiospores are just exposed spores. So the reason that we care about this or worry about this in a clinical setting is because sporangiospores are maybe a little bit easier to contain. We might be able to stress about them a little bit less, but depending on how far along the infection is and if we can get rid of those organisms. Where conidiospores, you touch that thing and you try to remove this from your patient, and your patient's full of spores. So that's something that we worry about as well. So conidiospores can come in different shapes. They can come in different weird little shapes depending on what the genus and species is, but conidiospores are exposed, sporangiospores are enclosed. So an example of a conidiospore is aspergillus. So that's one of our note cards. I don't know if that's ours today. I don't think it's today, but aspergillus is a black mold infection. So we'll talk more about that, about black mold and how many people in there have black mold in their homes and why we worry about that. But also penicillium. So remember I drew the little cartoon always about the penicillium mold, and penicillium was the accidental discovery of Alexander Fleming, where he found that it would kill the bacteria, but they have conidiospores as well. So we'll look at those in the microscope. So here's aspergillus, an example. This is like a bigger vesicle. I don't care that you know that, but just so you know what they look like in the microscope. And then here's like a rhizopus, for example. The sporangiospores are inside, the conidiospores are outside. So colony morphology of a mold. So they look like what grows on strawberries or whatever. They look like what a traditional fungal infection you might think looks like. So they are if you look at it like even under a magnifying glass, you'd see they look like little tiny like grasses or little tiny trees. They have hyphae that come up and they look thick. And if you go like this and dumped it, which you never should, but if you were shaking it, you'd have trillions of spores everywhere. So that's what they look like when you grow them on agar, and we'll be able to see that in the lab. So this is what molds look like. So if you could tell like the spaghetti and meatball example, they'd have more spaghetti versus meatball ratio, and depending on whether these are sporangiospores, but conidiospores might look more polka dotty, but basically you'd see all this hyphae everywhere. So dimorphic fungi, there comes in two types of shapes. So they might be a mold or they might be a yeast depending on temperature. So there's only a few examples of this. There are only a few genus and species that are dimorphic. 1, we really worry about every day, all the time. 3, we don't really worry about where we live because they're really prominent in warmer, more moist areas such as the South and maybe the southern East Coast areas that they worry about dimorphic fungi a lot. But there is a type of dimorphic fungi that we've been worrying about more and more in California because it has been creeping up from Mexico, where parts of Mexico have had the problem, but now because of temperature changes, it has kind of come up more towards in California, and we've had more instances of dimorphic fungal infections. So here's at 30 degrees, 37 is body temperature, 30 degrees is colder than body temperature, so they can survive out in the environment, right? When it gets a little bit chillier, 98.6 is not year-round temperature in a lot of places. Usually it's a little bit chillier than that, even if it's 80 degrees or something. So they are a little bit more versatile. And if you think about that type of infection also in the human body, like where might they live, or what morphology might they exist in the human body, that kind of thing. Okay, so here's the dimorphic fungi examples, and you don't need to know. I think we have as part of our note cards, we have Canidia, definitely as part of our note cards, but maybe Coccidioides. So the reason I can't stand some of the fungal names that are really gonna drive you bananas is because what does this word remind you of?
Cocci.
Cocci. What does Coccidioides remind you of? Yes, Clostridioides difficile, which they made those two terms way too similar. But this is has nothing to do with Clostridioides difficile, which is a bacterial infection, that's C. diff. And it has nothing to do with cocci, it's not a bacteria. This is a fungal infection. Yeah?
Can you pronounce the last one again?
Paracoccidioides.
You said Candida?
Oh, Canidia,
Candida?
Oh, I think I spelled that wrong.
Oh, okay.
Oh, Candida. Oh, that's Candida. But it is candidiasis. Why do I keep saying candidiasis? Because it is candidiasis. Let me come back to that. I'll come back to that after our note card today. Okay, so blastomyces, coccidioides, histoplasma, paracoccidioides, and candida is a candida infection, but I'm getting confused between those two terms. Let me come back to it. Okay, so dimorphic fungi under the microscope, these look like all kinds of weird things. We see some yeast with some buds. We see some little polka dots that maybe look like yeast. And then we see some hyphae that look like molds. So this is what they might look like in a warmer environment. This is what they might look like in a colder environment. But we would know these are not bacterial infections because of what?
Their size?
Yes, because of their size. Because under the microscope, we would know it looks like your patient has an infection, but they're huge. What could that be? It's a yeast. Okay. So here are some of our terms that I just went over. You guys can go over that. And then I am going to move on. Oh, and you know what we're going to do is do our mitosis versus meiosis. But let's do our note card really quick because why was I saying? Because it is Candida albicans. What was I saying?
Candida.
Oh no, I am all off. Canidia are these.
Oh.
Yes. Candida albicans is a yeast infection. Oh, so I need one note card. It's so small. So, this is Candida albicans. So say it 3 times with me. Candida albicans.
Candida albicans.
2 more times. Candida albicans.
Candida albicans.
Candida albicans.
Candida albicans.
Okay. So this is what it might look like. These are dyed with probably crystal violet, just to confuse you. But these are not bacteria. have you ever seen bacteria that look like this? Like, if you zoom in, never, you'll never get this kind of view of bacteria because we don't have that strength of microscope. And then also what you'll be able to see when you look under the microscope are these little buds. So a lot of them are reproducing if they have enough nutrients, and these little things are called buds, and that's characteristic of Candida infection or a yeast infection. I guess you should say fungal infection, but I'll tell you what I mean in a minute. So maybe under your note card, label magnification for sure, and then label like a bud, what that looks like. Candida albicans is a huge problem clinically. So I'll let you guys write that down and then explain what that is. Okay, so these are dimorphic fungi, and we do care that they're dimorphic. We do, because we do care how they're transmitted, but we also don't care. I mean, the reason that we don't discuss that very often is because we care what happens in the human body so much. So basically in the human body, they live in the yeast form. They look like a yeast. They're a human commensal, so every single one of us have Candida on us at every given moment. But they're opportunistic. So what does opportunistic mean?
They wait for a low immune system.
Awesome, yes, so they take advantage of an immunocompromised patient, and then they're very common nosocomials. What does nosocomial mean? Hospital acquired, awesome. Or maybe now the hospitals want us to say associated, so they don't get sued, right? So yeast, they're very round and very large, and molds, they have hyphae and they also have pseudohyphae. There's about 100 species of different Candida. Drug resistance we worry about a lot. So candidiasis is a yeast, and the name of a Candida infection is candidiasis. So you'll see this more and more with fungi in particular. They just use the name of the fungi and add the disease ending to it, but we'll go over that at the end. So thrush, have you guys heard of thrush before? Yeah. So babies get thrush. A lot of babies get thrush. I won't know what to say all of them. My daughter never got it, luckily, but a significant portion of babies get thrush, and it looks like white and creamy on the tongue, and it's a yeast. It's overgrowing yeast in the mouth of babies, and so usually what they do recommend is breastfeeding and keeping the breastfeeding surface clean, keeping bottles clean, keeping anything that they put in their mouth, keeping that clean, and it'll usually go away on its own. A yeast infection, so candidiasis of the vagina is generally called a yeast infection, and that's from candidiasis, and it's not sexually transmitted again. It's usually because of the pH balance of the vagina is off. So losing those lactobacillus beneficial bacteria can cause a yeast infection. And so as we get further on into the diagnostic section, we have three main, I don't know if three, we have a few main vaginal issues that are common, but if your patient comes in with some sort of itching, burning sensation, and you treat it as if it's bacterial, like with antibacterial cream or something like that, it can exacerbate the yeast infection from candida. So it can be very, very bad for your patient if you misdiagnose your patient with a yeast infection versus BV. What do you guys remember the organism that causes BV? Gardnerella vaginalis. Awesome. Don't forget that.
So the like environment of like the vagina, does that make something like?
No, that's a really good point. I guess you could say immunocompromised, but they're not really immunocompromised, but usually it's a pH problem. But that's a really good point. Yeah, so a lot of swimmers get this. I always worry about my daughter about getting either BV or yeast infection because she's a swimmer. We've been lucky so far, but because of the chlorine in the pool, so it could be all kinds of different things. It can be diet, it can be they got an illness, it can be they took, generally, I would say 8 times, this is just my non-clinical experience from when I worked in the hospital, 8 times out of 10, the patient has BV or yeast infection from secondary to taking antibiotics. So that's always really, if you're a female, even if you are a male, it doesn't matter if you don't have a vagina, because taking antibiotics wipes out your beneficial bacterial population, so it can harm the gut, it can cause ear infections, eye infection, all kinds of other stuff because you got rid of all of your commensals. So yeast infection and invasive, it's called invasive candidiasis, is in the bloodstream. That can lead to sepsis, which we'll talk about later on in the semester. Pulmonary, so it can cause pneumonia. And cutaneous is a skin infection, which is common actually. And usually it's because there's some sort of something going on with the skin. They have some sort of comorbidity, like for example, patients with diabetes that have mismanaged diabetes can have breaks in the skin and have exposed tissue, and that exposed tissue can get a candida infection. So that's very common. Or if your patient is laying too long in one position and that it gets moist and the skin gets compromised because of the moisture. Or also if you have a patient that has layers, and between the layers, if they're not cleaning very well, then you're gonna love your job. Basically, you're gonna love your job. Okay, I'm trying to think of how to say it. Okay, so you're like just watching me struggle. Everybody's like, just keep going, just keep taking. Okay. The reason I think of all of these things is like, it always brings me back to like my stories that my sister tells me of like all of her patients, of like the, like homeless patients or patients that don't have good hygiene, and the yeast infection that occurs is pretty special. Okay, let's move on.
Like the sores that like some people like can get, like those layers, like is that usually yeast infection?
So they will get a yeast infection, but the sores generally are just friction, and then because if they have any exposed skin and it's moist and it's, just there's yeast everywhere, and so there's yeast on our skin all the time, and so if it's moist and there's exposed tissue, then they'll set up shop and they can live there. Okay, so pseudohyphae and hyphae, those are just some ideas of what they might look like under the microscope. You won't have to identify the difference between those ever. I just wanted to show you what that looks like. Here's the colony morphology. I think this is kind of a better picture of what it might look like for yeast versus a mold. And that a bacteria, they don't look like that. They're not as convex, they're not as shiny. Also, you don't want to smell your bacteria, but you can smell it. As soon as you open the plate, you can it smells like beer, you know, you can smell it right away. So here's the difference between yeast and bacteria in the microscope. They're huge in comparison. Okay, and then we did our note cards and we're good to go. Okay. So we are going to talk about mitosis versus meiosis for the last few minutes, and let me have you guys get out your note cards. Yeah. Or, I'm sorry, yours.
You mentioned there was the one biomarker fungus that was floating up from Mexico. Do you remember the name of that?
Oh yes, it's histoplasma. Histoplasmosis is the one that's causing a problem right now.
That's the condition name?
Histoplasmosis is the condition name. Histoplasma is the genus. So there's different species of histoplasma, and the species is, or the name of the disease is histoplasmosis. The genus is histoplasma. Yeah, great. Okay, so I'm gonna have you guys come up and grab one for your table, and maybe if there's 4 of you at a table, come and grab 2 so that you guys can go in just groups of 2 to 3-ish. And first, what I'm gonna have you do is grab so just in a few minutes while I get set up, I want you to do our cell, eukaryotic cell, and all the organelles and the functions together as a team.
Oh, sorry. Oh, yes, she had you right here. Oh, she dropped. Yeah. She wasn't. Shoot, I haven't heard from her.
Oh, I'm so sorry.
Okay,
Oh,
encase things that are bad so that they don't affect the rest of the cell. They can encase things like proteins that are good and then add them to the cell.
That's like one of the things, yeah.
And the Biol proteins and whatever are floating around and you don't need to know about that.
Oh, yeah, you're doing it. You're eating, are you both? Or they're not really-
We're in it right now. I'm more in it.
How do you do that?
And they actually are.
Do you guys need one more minute? Or are you done?
We're good.
Good? Good? Okay. Okay, let's move on. Okay, so let's talk about mitosis. So in mitosis, remember we're talking about cell division. So we're talking about the cell cycle. So we kind of did this at 12:30 and went back over it one more time. But don't forget when you're talking about the cell, the cell cycle, you could be talking about one organism going through the cell cycle and becoming two organisms. Or you could be talking about one cell within one organism becoming two cells within that organism. The same organism. So single cellular, multicellular. Does that make sense to everyone? This is exactly what our skin cells will go through. Or this is what one individual yeast will go through. Yeah? Okay. Okay, so this is an organism with 4 chromosomes. How many do we have? 46. So we have 46. So we just giving us a simple of ease of whatever organism has 4 chromosomes, so we don't have to draw 46 lines. But it is a good idea for you to try these in different numbers. Maybe I'll have you guys try that. So we have 4. We go through interphase, we go through mitosis and cytokinesis. So through interphase, organelles replicate. Name all the organelles. We go through DNA replication, DNA polymerase delta. It looks like an S. It helps us remember that it's synthesis, DNA pol delta. And then it goes through proofreading, DNA pol epsilon is the editor. So after interphase, and when it's gonna line up on the metaphase plate, it's been replicated. So now we had 4, now we have 8. Those 8 are gonna line up on the metaphase plate and split off into 2 different cells, exactly genetically identical. Yeah? With me? Okay, moving on. So now we're going to talk about meiosis. And the reason that I bring it up later on in the semester, not later on in the semester, is 2 days ago. But the reason I bring it up a little bit later is because it's only in sexually reproducing organisms, and it's only in the organisms that have gametes. So they have to have some sort of sexually reproducing cell. And that sexually reproducing cell is a cell that will go off and has to meet up with another cell. Has to, I should not say that. Yeah, because sometimes they can haploid to haploid. So, okay, so let's just get to the bones and then hopefully it'll make more sense. So when we're talking about a mold, for example, and we're talking about one spore, the spore itself is a gamete. That spore itself is a sexually reproducing egg. And then in a human, there's eggs or sperm that are the sexually reproducing gametes. So these gametes can be combined with something else to make a full number set of chromosomes. So if you're if every other cell on this plant has, for example, no, let me not say that. This one has 8. So if this organism has 8 chromosomes, then their sexually reproducing gametes have 4. So it's the number is divided by 2, it's halved. So when one cell has the full chromosome set, that's called diploid. So diploid is a full chromosome set, diploid. And usually we represent that as 2n. That means 2 times the number of chromosomes. So that's a full set. Usually it comes from a father and from a mother. One comes from a father, one comes from a mother. And that's a diploid cell or a diploid organism. Yeah,
So a diploid is made from 2 gametes, right?
from 2 combined.
Yeah, so you take
2 combined.
one gamete from the mom, one gamete from the father, and then it makes a diploid.
Yes.
Okay.
Awesome. Okay, so then we have a haploid, which is just represented by the letter N. And this is half the number of chromosomes, and it's usually a combination of mother, father, but it's half the set. So when we're talking about the actual cell, the actual organism, all of the cells within the organism have a full set, or diploid. But if it has a spore or some sort of sexually reproducing gamete, it's generally haploid. It's half the number. But let's talk about it in humans, because humans it makes more sense. Fungi are really crazy. Humans, all of our cells, eyes, ears, nose, mouth, liver, lungs, kidneys, everything have the 2N, or diploid. We have every single cell in our body has half the number of chromosomes from your mother, half the number of chromosomes from your father. Except in sperm and egg. So in sperm and egg, they have half the number. And the reason is because half that number is gonna combine with the other half number to make a full organism. And evolutionarily, it made sense, because evolutionarily, how, just like Dr. Myers was saying at the beginning of class, when you have a combination of genes, it makes for a more successful organism over eons. Since the beginning of time, anytime that there's some sort of genetic combination or genetic variability, it leaves the organism less vulnerable to disease, to any kind of mutation or things that happen within the genome. So, okay, we're there? We're good? Okay, so if your organism has 8 chromosomes, for example, and the gamete itself has 4, when it goes through S phase and it goes through replication, now it doubles in the number of chromosomes, but they line up differently on the metaphase plate. So on the metaphase plate, they're not gonna go single file. They're gonna go to partners. So these partners will line up on the metaphase plate. And in meiosis 1, they're gonna split off into partners. So metaphase 1, they line up in here along, if you pretend there's like an imaginary line, the chromosomes will line up in partners. It's called a tetrad. They'll line up in partners. So then once they split apart, then these 2 are gonna go this way, and these 2 are gonna go this way. So they are separating out into their areas. So just like I wrote here, but this is metaphase 1, this is metaphase 2, where now they do the traditional lining up. And then they're going to split apart into their own cells. So now we have 4 haploid gametes. So what this one looks like if you compare mitosis and meiosis, mitosis here looks like here. So it looks like metaphase 2. Metaphase 1, these don't match, right? This one and this one doesn't match, where the tetrads line up along the metaphase plate. So these ones are gonna split off here separately, so we added this whole phase 1 that's different. Phase 2 is doing the exact same thing as mitosis. Now we have 4 different cells that each have their own haploid genome, half the number. So it depends on how these line up, and it depends on something that's called crossing over, but it depends on what these cells are gonna contain that made you you. So it depends on, you might have some characteristics from a grandmother on one side and some characteristics from a cousin on another side. And that's because of the way these line up and the way that they do something called crossing over, which we don't care about for the sake of this class. But these 4 can become sperm, but only one of these can become an egg. The 3 die in females. They become polar bodies, they're called. But we don't care about human genetics. We're talking about yeast and molds at the moment. So what I'm gonna have you guys do is that, do you guys have any questions about that? Okay, I'm gonna have you guys practice. So how do you feel if I erase this? You're good? Yeah? Okay, I'm gonna take a picture of it so I can draw it again on Thursday. So now I'm gonna have you guys go through mitosis with an organism with 8 chromosomes. So go ahead with if you wanna come up and grab another color, if it helps you to keep track of the chromosomes, you're welcome to do that. But get together with your partner and draw the whole cell cycle in an organism that has 8 chromosomes.
Mitosis or meiosis?
Mitosis. So go through mitosis.
Oh, that's all good. I'm not proud. Okay, so we got this. Yeah. Oh wait. That's why meiosis happens. So we're gonna.
It's kind of true, like.
What? Epsilon. Mm-hmm. Yeah, like a little something.
Oh, just overall. Where? Oh yeah, yeah, right. So oh, I was thinking this is mitosis. That would just be 2, but it would be 8. Yeah, this was so it wasn't flat. We're on the 4 second. So it's what the insider is. Okay. Alright. Nice. Is there an actual word?
Yeah.
Is this a page? This is post it.
Okay, do you guys need me to do it on the board, or you feel pretty confident?
Okay. Doesn't seem like it's, it seems like it's more than 2 mitoses. It's just the same thing for like just these stats.
Okay, how do you feel about it? You guys want to do it on the board, or you feel good? 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 Okay, so with this one, we did organism that has 8 chromosomes. So draw one that has, I know it'll be a lot of drawing, draw one that has 12 chromosomes. So draw meiosis or meiosis, however you want to say it, for an organism that has 12 chromosomes. You're welcome to get 2 colors if you use straws.
Yeah, that's right. That's right. Oh, give or take a piece. Do you work at a hospital like the same day? Oh, it's not a company. Fair. What age range? Like for kids or like just oh, gotcha. Oh, I'm wondering. Oh, because we're doing, that's, alright, there's not cleavage, but just, yeah, yeah, that's gonna be, well, they're gonna be on that line. So can I say that?
Is office downstairs?
Yeah. She should be. Yeah.
Because we're supposed to build in Thursday morning. I just wanted to talk to her
Oh, for what?
She's I think that they're doing like a lab review, but they're doing like a new hire, so
Oh, sorry.
so we're just gonna like sit in, and she's gonna have like stuff all laid out.
Oh, yeah.
And they're just gonna be guarding like stuff in the lab practice.
Oh, awesome.
So I have to be here pretty early on.
Oh, I just like, okay, cool. Oh, yeah, yeah, she should be downstairs. You're welcome to go down. Her office is, if you're looking at the lab door, it's right, the first door right behind you.
Okay.
Yeah, there. Yeah. Okay, so how does that, do you guys, does that make sense?
But that's for people too, right?
So, 12. There would be, at that point, there would be 24.
Okay.
Yeah.
So
So down there, they would have. It starts off with 12 individual lines, and then it makes the X, so that's when you have the 24.
So what we're drawing is the human version. Is that the Mongol version?
Well, they do the same thing.
Okay, this looks the same.
Yeah, it
Okay.
looks the same. Yeah.
Yes.
So what you're lining up on the metaphase plate are let's draw them as X's, 2, 3, so that's 2, 4, 6, 8, 10, 12. So that's meiosis 1, PMAT 1. And then 2 would be 6, because that'd be 12. And then in another cell, like that. Does that make sense?
Why are they in the?
So this is meiosis 1, where the tetrads line up. And then this is meiosis 2, where they line up here individually. And then you'd go back to,
Is this prophase of each of them?
this is metaphase. So that's metaphase where they're lining up along the metaphase plate like that. So then each should have 6. 1, 2, 3, 4, 5, 6.
Okay.
So then you'd have 4 with 6.
Yeah.
I'm so crisp.
Does that make sense?
Yes.
Back row? Let's draw it again.
So, I think we only have 2 cells here on this page. Because I only showed the ones to count and it cards up to the didn't have 4 cells.
Let's just draw it closer. So always either during mitosis, metaphase of mitosis, or meiosis 1. Let's not talk about mitosis. Let's talk about that separately. So if you have 12 chromosomes, you're gonna have 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. That's a normal cell. Now it's gonna replicate. So now, without drawing them anatomically correct, this one replicates, this one replicates, this one replicates. So now you have 24. Right? Yeah. So now, this is metaphase, because they lined up in tetrads, they lined up on the metaphase plate. Now they're gonna go their separate ways down the middle. So down the middle, you're gonna have 1, 2, 3, 4, 5, 6, but it's still replicated. It's still in its X form. I know I'm drawing it like a K, but it's still in its X form. Here you have 2 of those. So this is meiosis, or mitosis, I'm sorry, no. Meiosis, metaphase 1, meiosis, metaphase 2. And at the end of cytokinesis of metaphase 2, these are gonna split off into 4 cells. So here it goes to here, let's draw this around to here. 1, 2, 3, 4, and this one's gonna have 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. Because the yellow went to one side and the blue went to the other side here. Yeah.
What's the last? This is meiosis or metaphase 1, metaphase 2.
And then that's just after cytokinesis. They split up. So this is how they line up on metaphase plate. And then when they split apart after telophase, cytokinesis, then there's 4 different cells. So your original organism had 12, and your haploid has 6. Haploid gametes has 6.
And the haploid would be another haploid.
Yep, this would be meeting up with another organism who had another 6.
Okay.
And then it would make a new 12.
Yes.
Yeah? Haploid is describing the number of chromosomes. Gamete is describing the cell itself.
Okay.
Yeah, so you'd say a haploid gamete is like a half number of chromosome sex cell.
Okay.
Yes, yes, yes. So one more thing I want to say about that. Oh, Dr. Buglino, I was saying about the stem cells, but don't anybody else stress about it. I'll just tell you specifically, is that I'm gonna confuse everybody if I tell them this, but I'm just gonna throw it out there about the stem cell situation, is that when you have the original cell that gives birth to the gametes, that's the stem cell. So the stem cell of sperm cells or of egg cells has the full set of chromosomes and will continue to have the full set of chromosomes forever. They'll replicate on their own, and then they'll do something separate and go through meiosis. They go through mitosis and then they go through meiosis, but it's the stem cell that makes those cells. Yeah, sorry, just throwing that out there.
So
Yeah,
the
stem cells are like mother cells. They can give rise, what a stem cell is it gives rise to its own self plus another cell type, which is what makes them unique. So like a basal cell of a skin cell can give rise to new basal cells, and also basal cells can give rise to keratinocytes. So it has 2 jobs, and that's what makes it a stem cell, is it can give rise to other cell types. Yeah,
So are you saying that the spermatogonium and the oogonium are stem cells?
stem cells. Awesome. Cool, okay, so get cleaned up right away in the lab because we're gonna like INVIC right away. I'm hoping we can, I'm not usually a leave early lab kind of person. I like to stay to the very end because we have so much to cover, but I'm hoping